Inside the womb, human development operates as a high-speed construction project that extends far beyond simple physical growth. Memory and cognition are already active through transnatal learning, the process where sensory data gathered in utero persists and carries over into the postnatal world. We can measure this continuity in newborn behavior. Shortly after birth, infants show a clear preference for the specific intonations and rhythms of their mother's native language, easily differentiating it from foreign speakers. Infants also demonstrate a preference for specific stories. If a mother reads a particular book aloud repeatedly during her pregnancy, her newborn will react more calmly to that exact story than to an unfamiliar one. Because a fetus is already processing and storing complex sensory information, a significant biological question remains. What physical machinery is being built in real time to capture this data? Birth marks the moment the brain's first invisible construction project is finally put to the test. This 5-week-old embryo shows the earliest stages of the nervous system. Even at this stage, the head region is already disproportionately large as the neural tube begins to form the rudimentary brain. Around the 9th week of gestation, brain development enters a phase of exponential acceleration. This is the start of neurogenesis, the rapid production of the core cells that comprise the nervous system. The velocity of this process is immense. During this window, the fetal brain generates between 250,000 and 300,000 new neurons every single minute. By the end of the second trimester, this rapid multiplication has produced roughly 200 billion neurons. The first 2 trimesters represent a high-speed hardware manufacturing phase. The body produces the raw physical material of the mind at a scale that will not be repeated at any other point in the human lifespan. Rapid neurogenesis creates a physical dilemma. Billions of new cells must be packed into the highly restricted, finite space of the developing skull. These illustrations track the early fetal brain. Notice the top row. Between 25 and 100 days, the surface remains smooth and rudimentary. To maximize surface area, the brain folds deeply upon itself. You can see the complex wrinkles in these 8- and 9-month illustrations. This folding process is a core example of neuroconstructivism. The principle suggests that brain architecture is not built in isolation. It is actively shaped by the constraints of the body and the surrounding environment. The final physical architecture of a human brain is a direct negotiation between explosive cellular growth and the immediate physical environment of the womb. Because the brain builds itself at such a high speed, it is extremely sensitive to external chemical signals. When a mother experiences stress, her body releases cortisol. This hormone crosses the placental barrier and enters the fetal environment, directly interacting with developing neurons. Other environmental factors also alter the construction process. Poor maternal sleep quality early in pregnancy is linked to changes in the volume of the hippocampus, while nutritional deficits, like a lack of folate, can restrict the expansion of the frontal and temporal cortices. These maternal experiences act as permanent architectural instructions. They alter the development of crucial memory centers like the hippocampus and amygdala, shaping the brain's baseline before the child is even born. Life before and after birth represents 2 distinct phases of neurological development. The prenatal period is dedicated to building hardware, generating massive quantities of raw neurons. The postnatal phase focuses on wiring them together. Sensory input from the outside world builds trillions of complex connections, the software, between cells produced during gestation. A child's ability to wire these postnatal connections is dependent on the structural integrity of the hardware built in the womb. These 9 months are the brain's most vulnerable window of development. The womb is the first classroom, the moment where biology and the environment intersect to forge a lifelong cognitive trajectory.